Rise and shine. A very good morning to one and all present here. Hope everyone are safe and good. This is Neha, third year Biotechnology department. Let's get started with a quote. A great attitude becomes a great day, which becomes a great year. Since everyone is here, let's get started. To welcome you all, I would like to call upon Dr. Rajeshwari Hari ma'am, HOD of Biotechnology department.
Okay. Uh, Good morning, one and all present here. On behalf of Biotechnology Department and Dr. M.G.R Educational and Research Institute, I take the great privilege of welcoming you all for the technical talk entitled Biotechniques by Dr. Prakash, who is Assistant Professor and Head of the Department of Biotechnology, Vels Institute of Science and Technology. As a biotechnology students, you are all supposed to handle various equipments and instruments. So you have to know the standard operating procedures of these equipments, and definitely I think this talk will help you in understanding the standard operating procedures of these equipments and instruments involved in the research of various biotechnological investigations. So definitely this lecture will help you to understand and update the knowledge on various techniques involved in biotechnology field. So please carefully listen the talk and update your knowledge, which will be useful to your future academics as well as research. And once again, I welcome the speaker as well as the participants for this technical talk, and listen carefully and update your knowledge and enrich your knowledge in the handling the different equipments and instruments. Thank you. Thank you for the opportunity given to me to welcome you all. Thank you.
Thank you so much for your welcome address, ma'am. To welcome our special guest, I call upon Ms. Sandhya ma'am, Assistant Professor of Department of Biotechnology.
Good morning to one and all present here. It's an immense pleasure to invite our chief guest, Dr. B. Prakash, Associate and Associate Professor and Head of Department of Biotechnology, Vistas. So I would like to give a introduction about our guest today. Sir has about 18 years of teaching experience and 7 years of research experience in the field of biotechnology. The area of specialization of the Sir is animal biotechnology and medical biotechnology. Sir has given more than 10 publications in well indexed journals and has received about 4 awards for his immense publications and for the posters and paper presentations. Not only that, Sir has about 3 funding projects and has completed in a successful way. And I'm very glad to be the past student of Vistas, and I welcome you all for the department talk. And I welcome you, sir, for giving us a very will be giving you a very informative talk to our students, and we are very happy to receive a talk from your side. Thank you.
Thank you, ma'am. The most awaited moment, a special guest, that is our chief guest, Dr. B. Prakash, sir, Associate Professor and Head of Wales Institute of Science and Technology and Advanced Science, will be presenting now. Over to you, sir.
Thank you, all of you. So, first of all, I should thank the Dr. M.G.R. Education Institutions, as well as Head of the Department of Biotechnology, and my dear colleagues, friends, and my dear students and participants. So, first of all, once again, I thank you all for giving this wonderful opportunity for me. So, today, my topic is Biotechnics. Actually, why I have chosen this topic, I'll tell you one by one. Uh, okay, now I just share my PPT. It will be help you. So I think you all can view my slides. Is it now? So, but okay. Anyway, I did not expect that it will be an interaction session, but fortunately, it will be in live, so I cannot able to interact with you. I think so. So anyway, that's not a problem. Even though I just want to give some ideas. I think you all can view my slides. So today my topic is a basic understanding of biotechnics. Actually, biotechnics. I don't want to go directly in this about the biotechnics. Okay, before going for this biotechnology or my presentation, I just want to give some basic about the curiosity. So ideas comes from curiosity. This is told by Walt Disney, and as well as when you're curious, you find a lot of interesting things to do. If you have the curiosity, you can do a lot of things. To be frank, without curiosity, you cannot survive not only in this field of biotechnology, not in all the field, because we must know what's happening, why it's happening, how it's happening. Everything based on the questions only we can able to know the basics of basic understanding of each and every technology. So here, before going for this curiosity, I just want to ask you about these things, because if it is an interaction session, it will be very helpful for me. It's easiest for me, and easiest for me. It will be uh, how can I say, interesting one. But fortunately, of course, it's not a problem. Okay, I think if I'm asking about the bike, I think here I'm showing the KTM bike and mobile phone and one jewels and all. If I'm asking about the what is the make and model of this bike, anyone can answer. Most probably the boys, most of the boys, even some of the girls also, they know very well about the bikes and the what of the types of bikes and how what is the CC, what is the make and model, and what is the cost of the bike. Everything we know, because of the curiosity is very high in this model of bike, mobile phone or jewels and if I'm asking about the jewels for the girls, they can able to tell more than that what I know, because you are all very curious to learn what are the updates are there, what are the new model of the mobiles we have. Even if I'm asking about the what kind of mobile phone you are using, what are the basic models, what are the features are there, if I'm asking like that, anyone can answer. You all, most of the people are know so many things about the mobile, because when you are going to purchase a mobile, we at least we do research for 1 month or 2 month, okay, minimum. If for example, in case of boys, if they want to purchase one bike, if they want to, even if they don't want to purchase the bike, if you any new bikes launched by some companies mean, before that they are having very much curious to know about the bikes. So but I know, but that much of curiosity is lacking in their laboratory. That's the main problem of the students. Okay, so I just want to ask you one question: how many of you know the make and model of the instruments which you have used before in your laboratory, even it's maybe a plus two or your UG or whatever it is, in your first year or second year, or even if you're doing PhD also. So many of you, they do not know about this make and model of the instruments. You know how to operate the instruments, what all the instruments are there, that you know. That's not a problem. I know very well that things. Even some of them are may not know that also. But even though the make and model of the instruments, why I'm asking this make and model of the instruments, that is very essential here. I'll tell you later one by one. For example, take an microscopy. I have a lot of questions in the microscopy, because I know, because the basic fundamental things in a lab, because you all may know, you may expect a lot of things from me. Latest trending technology, how to calibrate, how to SOP and all standard operating procedures and all. But I don't want to tell you anything right now, because within this 1 hour, I cannot able to teach you all these things, because it's all available in the internet or Google or your faculty members, though they are know more than about me for this all the instruments and technology and all. But the students are lacking the interest lacking that curiosity. I just want to motivate the things because within the 1 hour I cannot able to teach all the methods or technology or whatever in C, blah 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 etc. So I can able to motivate you curiosity. I can able to induce your curiosity. That I can do. That only I'm going to do here. Do it here. I don't want to take. I don't want to tell about the flow cytometry or sequencer or something blah blah etc. Okay. I just want to ask you the some simple basic things. We are missing all those things in your UG or PG level. Okay, even if we have did a lot of things in your lab UG or PG level, but even though we missed something, if I'm asking one questions, what do you know how to calibrate the microscope? And do you know the meaning of the magnification? You know the meaning of the magnification. Everybody know the proper theoretical part of the magnification. Okay, but my question is, how many times we can able to magnify the object or specimen by using the student's compound microscope? This is my questions. So most of you, they may not know. If you are telling the 10 times or 100 times or 40 times, 90% of the results are come like this only. But actually it's not a right way. Actually, you know the parts of the microscopy, even in your plus 2 level you all know about the my plus 2, on plus 1 onwards you know about the microscopy. So even though I'll tell you, even though to be honest, everybody has to accept that we may not correct the magnification. We may not know the magnification because how many times we are able to we are can able to magnify the object or specimen in this students or compound microscope which you are used in your laboratory. Already you are used in your experience now. I'll tell you now. In case of there are three type of lenses are there in this microscopy. That means the in objective lens, 10x, 45x, 100x. That you know. The another kind of lenses are available in this eyepieces. Eyepieces also there is a lens. You never seen that eyepieces are having one number. It's maybe in 5x or 10x or 15x. Most of the time 10x or 5x maybe there. It depends upon the laboratory or microscopy. Okay. So the total my magnification is uh, before I tell you, I want to ask you the questions, but I cannot able to ask you questions right now. So I'll tell you, 10x means it's not an x, it's a multiplication. Actually, most of the students are they don't know about this x meaning of the x. It's a multiplication. So eyepieces as well as the objective lenses. For example, in case if I was keeping the specimen in objective lens, 10x objective lens means 10 into 10 times. That means once if we can able to magnify the object by using the objective lens 10 times. After that, we are going to magnify again 10 times by using eyepieces. So the total magnification size is 10 into 10. Those who totally 100 times we can able to magnify the object or specimen by using the student's microscope in case we are using 10x into 10 eyepiece, 10x a lens of eyepieces and 10x lens of objective lens. So in case of if you are using 100x oil immersion mean, we can able to magnify 1,000 times. That is the right answer. I know most of you people, to be honest, we have to accept these things where we are missing. Actually, to be frank, now it's a learning stage. So you can learn all these things from the basic onwards. So don't expect to do all these major techniques from now onwards. Okay, first of all, you have to understand the basic things, and then only we can go for the major one. Okay, so and also we must know the basic current updated models, and also most of you may not calibrate the eyepieces and objective lenses because even the microscope itself we have to learn a lot of things. That's what I try to say here. We may think like microscopy is a basic instruments everywhere is available. Why we have to spend time for learning all this kind of microscopy and all? But it's not a right way. Okay, the calibration of the microscopy it's using the ocular lens as well as the stage micrometer. There are two scales are there. By using the stage micrometer scale as well as the ocular micrometer scale, we have to calibrate the distance between the ocular lens scale. Okay, after the calibration only we came to know the distance between the scale, even the two scales. So then only we can able to measure the total size of the specimen or species. Okay, that's the basic things. I maybe some of them may do, some of them may know, and most of few because I'm having a lot of experience. In my experience, even my students also having this kind of trouble. They don't know this kind of basic things and all. So you try to learn this kind of small things also. From here only we can able to go the next level. Okay, likewise the pH meter. So pH meter calibration is one of the biggest task for each and every person. Even if we are PhD scholars, they have struggle by calibrating this pH meter. So you may think, if we nowadays we never check the pH when you are preparing the media and all. So actually calibration is a simple methodology, but even though we don't know or we don't have that experience for calibrating this kind of instruments. This is also a basic one for the calibration. So we have standard buffer solutions we used to have in our laboratory, like 4 pH, 7 pH, and 10 pH. So we have to set the pH based on the by using the standard buffer, and we have to adjust the temperature as well as the pH. Then only we have to go for pH analysis of your samples. Okay, so this is the one of the major and important instruments in our laboratory. But most of you never mind about they may never consider this kind of pH meter. It's you may think it's all small things. So I don't want to learn these things. I don't want to check the pH. Likewise, we are doing the practicals in our laboratory, so we are missing a lot of things by from the pH because the small changes, small adjustments in the pH also affect the total results of your the research. So each and every stages, even each and every steps is important for us to get the results, proper results in our research, or even the practical also. So likewise the micropaper there are four. So you I think yes, of course it's a, most of the time we may not have the experience by using the micropaper, because micropaper the basic operation, most of the people they may not know. There are two stages are there: light and smooth plunger plunger actions, and the locking mechanisms, and the how to keep the micropiper, and how to set the volume. All these things, there are so many things are there to learn about this microscope, forward and reverse pipetting. There are so many things are there, even though students they may not know. I know, that's why I suggest you to learn from the basics onwards. Micropipetting is the one of the essential things in our biotechnology laboratory, because we are going to use the 1 microliter or zero point 0.5 microliter of samples going to play in our laboratory, because the enzymes, restriction enzymes, or DNA polymerase, or a lot of enzymes, primers and all in our genetic engineering laboratory. We have to take zero point 0.5, zero point 0.2, or zero point 0.1 ml or something, that much of small quantity of the materials we have to use. Okay, if you are missing something you cannot able to get the results. So the standardization and calibration and experience, hands-on skill, is all very very essential in this field of biotechnology. So nowadays the nanotechnology has came, so it's more than that. So we must be very careful in this micro level things, because calibration and this kind of handling, we are I know that in India we are very poor in hands-on training, handling mechanisms, as well as we are lazy for learning this kind of small things and all. So that's why we are not getting the results in a major practicals or major research and all. So once we are doing the research or practicals, mean we must know where we are lacking, where we made a mistakes, that we have to know, because that's what is called the standardization of the practical. Even first time if you are doing some practicals, maybe we cannot get the results even the first time. So but we must know where we may made mistakes, that we can able to know by if you are having the skilled technology, then only we can able to get the results properly. So these are the latest micropapers. It's available nowadays. 8 channels, 12 channels, 16 channels, and sometimes 48 channels are also available. 32 channels or 48 channels. These are the latest trending of the micropaper. So at least we must know, if you don't have the if you can't able to afford this kind of micro, but at least we must know what is the micropetting, what are the technologies that are developed right now, because the education system as well as the industrial, there are a lot of distance between the industrial as well as the education background. Okay, education institutions, what we are teaching in our college or laboratory, it's all far away. It's a very old technology only we are teaching in our classrooms. But if you want to learn, if you want to work in our industry, they are totally the different sector, different kind of instruments, different type of methodology they are adopting, different type of latest technology they are adopting in their industries, or in research institutions and all. So we must update ourselves at least in theoretical, or there are a lot nowadays, there are a lot of googles are there in our hand, so we can update each and every seconds what kind of the instruments updated instruments are available in this, what are the things we are using nowadays in our laboratory. So likewise, we have to go the next things, DNA and RNA, DNA, RNA, and protein. Okay, so before that, I want to ask you one questions. You know the basic alphabets in English. There are 26 letters are there. By using the 26 letters, we know how many stories we can able to write. Actually, n number of stories we can write. This is infinity. That means we cannot able to tell this many stories where only we can able to write by using this 26 letters. Likewise, the evolution, by nature, they giving infinity type of species, just because of the arrangements of the amino acids or proteins on or nucleic acids or whatever it is, DNA bases or amino acids. Okay, based on the number of amino acids, based on the arrangements of the amino acids, the nature will differ. For an example, I'll tell you one example here: egg white yolk versus snake venom. So, for example, you take egg white yolk. Egg yolk, they comprise 48 percentage of water and 16 percentage of protein and 32 percentage of fat. And some minerals, some vitamins are there. Its white consists 88% of water, 10% of protein, and some other minerals. Likewise, the snake venom also is a complex mixture of enzymes as well as the protein. Both are more or less similar. Both are made up of the enzymes as well as the protein molecules. Both are made up of the amino acids. The 20 essential amino acids may be there, along with some minerals and ions may be there. Okay, but the difference between the snake venom as well as the white yolk, you know the difference. It's a snake venom is a toxin for human being, as well as the egg white yolk. It's good for health. It helps to induce our growth of that uh, plant or animal or whatever it is. Okay, so what is the difference between these two proteins? Both are protein, both are made up of the amino acids. The only thing is the arrangements of the amino acids. Alanine, for an example, alanine valine using isolation. This one kind of amino acids that we call as protein or enzymes. That enzymes or that protein, it's help for induce the growth of that animal. But at the same time, alanine valine using the small alteration between the amino acids, the same nature of the similar to the protein, but the only difference is the amino acid arrangements. These are both are contain the same amino acids, but the only the thing is arrangements of the amino acids in the snake venom. But it will be toxin for animal species. So, so this is about the evolution theory. So now let us talk about the bank robbery. It's a small an example, or for interesting of this session. So that's why I talking about this bank robbery. Okay. So what we have to do when we go for bank robbery? We have to plan for bank robbery. Okay, just for fun, I'm talking about this topic. So after that only, I'm going to talk about our why I am telling all these things. Okay, so before going for the bank robber, so what are the things we have to know? We must know the building architecture, what kind of materials they are used in this building, that bank, what are the security system, what are the types of CCTV cameras, security peoples are there, what kind of guns, metals they are having, what kind of locking system they are having, what is the lockers, where it is the locker, where the currencies are there, where is the jewels are there, how they are, what are the security system are interior and exterior, and what is the timing we have to plan. Everything we have to plan, and everything we has to know, then only we have to plan accordingly to rob the bank. Without knowing this kind of basic things, just like simply we cannot go and take the money from the bank. Okay, it's not the easiest way. So without planning, what will happen? Everything will be collapsed. So likewise, so for an example, I just to give you another example. For example, if we are taking the wall of the bank, the wall consists of bricks, cement, soil, etc., etc., etc. So they consist of the bricks or soil or whatever it is. Okay, if you want to demolish the bricks or wall, we need some weapons. We need some materials, or not a weapons, but some materials. Okay, So likewise, if you want to break that window or door or except whatever it is, if we need a welding or welding machine or something like that, we need. So this kind of specific instruments we need for breaking that wall or gate or whatever it is, for taking the money from inside the locker to outside the bank. Okay. So so 1 second, guys. So 1 second, guys, I'll just. Yeah. Hello. Yeah, I think you can hear me. So these are the instruments. We must know what all the basic things are there consist of this wall and our gate or grill or door or locker or whatever it is. Based on that things, we have to decide what kind of instruments, materials we have to take it for these things. Okay. Then the plan. We need a plan. So likewise, now we come for the isolation of the DNA, RNA, or protein. Okay. Isolation of DNA, RNA, and protein. Where it is was located, the DNA, it is located in the inside the cell, inside the nuclear plasm, or in case of eukaryotic cells. In case of prokaryotic cells, no need of worried about the nuclear plasm. But the cell is consists of what all the things are there in cell. The cell is covered with cell membrane. So first of all, we must know about the cell membrane. What all the chemical components are there? Cell membrane is made of what? What kind of materials are there? The cell membrane is consists of phospholipids and proteins, integrated proteins and fat, carbohydrates, peptidoglycan, etc., etc. So we have to think about this kind of chemical substance. Next, what are the stages are there? Cytoplasm. Cytoplasm, it consists of proteins. Most of the times, mostly it was contained water as well as the protein molecules, as well as it's contain lysosome, vacuoles, lot of minerals, carbohydrate. There are so many internal organelles may be there. Okay, lysosome we used to call as suicide body. So it was very dangerous one. Okay, once we remove lysis the or rupture that lysosome vacuoles, it will destroy the DNA also. So we need a DNA with safe manner without disrupting the DNA. Okay, so what are the mechanism we can able to adapt here? So that we can see. So there are so many methods are there: alkali lysis, enzymatic lysis, and physical method. So based on the laboratory conditions, based on the availability of the materials, based on that cell nature, in case of plant cell, animal cells, the chemical composition of the cell wall, cell membrane is differ from each and every type of cells. So we have to decide what kind of mechanism we can able to adapt for extracting the DNA from the cells. So we have to take this DNA. DNA is located inside the nuclear plasm. We have to take this DNA without disrupting the DNA or RNA or protein in the safest manner by breaking all the cell membrane, cell wall, nuclear plasm, and lysosome, vacuoles, phytophenolic components are there inside the cells. So without they may all disturb the DNA. So we have to avoid all these things by using buffer solutions. So buffer solution helps to maintain the pH. So we can avoid all these things. So these are the things I just want to share with you. So now let us talk about the preparation techniques and modern biological techniques. Because I already told you, whenever you use in a laboratory, whenever you do some practical or workout in a laboratory, we must know each and every about the instruments, about the make and model. What is the latest models are available right now? So everything we must know. Already I told you in these aspects. So now what are the basic, some of the basic technology we used in our laboratory? For example, cell and tissue culture, on centrifugation, chromatography, gel electrophoresis, spectrophotometry, polymer chain reactions, DNA sequencing, immunoassay, DNA cloning, microarray. This is the basically we are doing in our UGR PG level in each and every colleges. Even M.G.R University, you peoples are having a lot of very big instrumentation lab, centralized instrumentation laboratory we have. But I don't think so. You have visited in a laboratory or not? I request your ma'am to take you all the places. So now let us be see one by one cell culture techniques. So cell culture, I think, yeah, there are three type of cell cultures we may do in our laboratory: plant tissue culture, animal tissue culture, and microbial cultures, and all. So more or less all are similar. But the thing is, the media requirements is differ from each and every cells. So based on the media requirements of the cells, we have to provide a composition, chemical composition as a nutrient medium for growth of the plant in a sensitive manner. Microbial cell culture is for very easy compared to animal. Animal tissue culture is very easy compared to plant. Likewise, there are a lot of differences there each and every tissue cultures and all. Okay, so tissue cultures, cell culture, obviously in your practical, you may know these things and all. But even though I just tell you the outline, as a tissue culture is the growth of tissues or cells separate from the organisms. This is typically facilitated via use of liquid or semi-solid or solid growth medium, such as broth and agar. Tissue culture commonly referred to the culture of animal cells and tissues. So here, there are different type of mediums we are using now: broth medium and solid medium and liquid medium, semi-solid medium. Why? What is the difference? Why we have to go for semi-solid medium and solid medium? What is the difference between why we have to choose all these things? This kind of different type of mediums we have to use. These are the questions we have to rise up when you're doing the practicals or when you're doing research, because then only we can able to choose the right choice or right option for your cells, for your interest of your own cells. Okay, for that we must know some of the basics. Without basics we cannot decide what kind of mediums required for this kind of plant or animal or microbial cells. Okay, so likewise, the centrifugation, of course, the basics of centrifugation, but currently there are 1 lakh ultra centrifuges are there, are 1 lakh or 2 lakhs RPM and all there. The cost analysis, it's maybe comes under the crores and crores, nearly 1 crores. The amount Indian currency, I'm telling you, talking about the Indian currency, it may comes around 1 crores of the cost of the instruments are there. And also latest lot of technologies are adopted by here. So the but even though most of the time we may not know the basic principle of the centrifugation is the basic principle of the centrifugation is nothing, where the centrifugal acceleration is used to separate the substance of greater and lesser density. So yeah, based on the molecular weight, we are going to separate the molecules by using the centrifugal force. Likewise, the chromatography is a collective term for set of laboratory techniques for the separation of the mixture. So the chromatography as well as the centrifugation, both are separating the chemical substances or any molecules based on the different type of factors. Okay, in case of centrifugation, this molecules are separated based on the molecular weight. Here, chromatography also more or less similar, molecular size as well as the molecular weight. So there are different principles are there for each and every technology that we have to understand. Then only we can able to go for this kind of techniques. Even it's maybe paper chromatography or gel chromatography or gas chromatography, whatever it is. Okay. So likewise, you take an example of gel electrophoresis, because I think most of you may have the experience, or due to this lockdown period, you may not do all the practicals, maybe or may not be. But even though if you are doing the practicals, we all know the movement of the DNA or RNA or protein. It depends upon the size of the molecules. So here already we talked about the centrifugation and chromatography. It's also helps to separate the molecules based on the molecular weight or molecular size or etc. Okay, likewise here we are going to separate the DNA, RNA, and protein based on that molecular size. Okay, as well as based on that charge of the molecules that we know. I think most of you know very well about these topics. Even though once again I tell you, across the electrophoresis and SDS page, horizontal and vertical gel electrophoresis. Horizontal gel electrophoresis is used for DNA and RNA separation, isolation and identification. As well as SDS helps to identify the protein molecules. Okay, we can able to know the size of the protein molecules, and as well as we can know the size of the DNA and RNA by using agross electrophoresis. The movement of the DNA is based on charge of the molecules as well as the pore size of the gel as well as the size of the molecules. There are three factors that are responsible for the movement of the DNA or RNA or protein. Okay, first one is the charge of the molecules. Based on the charge of the molecules, the movement will take place, and based on the molecular size, the speed of the movement speed is depends upon the size of the molecules, as well as the factors are affecting the movements. The pore size of the gel, the what concentration we are using in our gel. In case of 1% gel, we may use it for plasmid or RNA. For sometimes the 0.7 percentage we may use it for DNA isolation, because for the difference is the size of the chromosomal DNA is very big compared to plasmid or RNA. So that's why we have to choose the best one. What kind of concentration we have to prepare for this average electrophoresis. Likewise, the spectrophotometry. Spectrophotometry, of course, is one of the uh, um, important technology we used for color to identify the color intensity, because the so many reactions, so many chemical reactions takes place inside the test tubes and all. So the concentration of the proteins or vitamins or minerals or enzymes or whatever it is, we can able to identify by using this kind of spectrophotometry. It's just simple logic. Based on the optical density, we can able to measure or we can able to differentiate the input and output. The based on the optical absorbs the percentage of the light intensity absorbed by that specimen. So based on that, we can able to tell calculate the concentration of the uh, chemical substances which is present in this sample, that we can able to tell by using the spectrophotometry. There are a lot of types are there for new updated versions of the UV analyze, UV visible spectrophotometry, ordinary colorimeter. There are so many techniques are there, to so many latest advanced models also there. You just update yourself by using Google or in the making industries and all, because so many industries, not only spectrophotometry, even the flow cytometry or sequencer, the companies they offer internship or training and all. So you try to get the training program or internship from those kind of instruments manufacturing companies, because they are the right person for giving the training program for that particular technology. In case of Illumina is one of the best methods for DNA sequencer, best instruments for DNA sequencer. So those companies are offering internship for the students, as well as the after completing your PG, you can get the training program there. Then after that, easily you can get the job. Okay, actual internship or hands-on training is you can get it from there only. So likewise, the polymerase chain reactions. Now there are the methods of polymerase chain reactions nowadays. When I was doing my PG, PhD in the 220 years back, we used to call only types of PCR. Only one type of PCR may available there. One methodology, that means either PCR or reverse transcriptase PCR. In case of RNA virus, we have to go for RT-PCR, or else we have to go for polymerase chain reactions in case of DNA. But nowadays there are so many technology, though so many developments that takes place in this PCR mechanisms. As a biotechnology students, we must be very strong in this PCR techniques, because nowadays nested multiplex PCR same time simultaneously we can able to amplify more than 10 genes in a single polymerase chain reaction machine. Okay, that kind of mechanisms are developed nested PCR. There are so many types are there available nowadays. So we have to go for Google for us to study all this technology, because I already told you for this within this 1 hour I cannot able to teach you all type of technology, all type of latest trending instruments and all. But I can just explain what are the methods we have to do, how to learn all these things. That only I can able to teach you now. So that's why I tell you. So likewise, the DNA sequencer. DNA sequencer is one of the major techniques we are using now. So you must try to get the training. I told you, try to get some. You have to find out the company, manufacturing company of the DNA sequencer. You get the training program there. You try to get. So you will get job easily. As well as I want to tell you one thing. You don't need to expert in all the technology. No need to I don't go in this broader level. Okay, just because everybody cannot be expert in all the time, all the technology. Even myself, I cannot be expert in all the type of technologies. I can able to know only, I can able to in my specialization is some narrow field, some animal or microbial cells and all. Okay, so likewise, if you have to choose any one of among these techniques, okay, you try to be strong on the particular techniques. But you must know the basics of all the remaining techniques, but you must be an expert on the particular techniques. Okay, so based on the particular techniques only, you can get your jobs, or you can get a jobs in industry or research institution or whatever it is. Okay, likewise, immunoassay by using the ELISA reader, I think antigen antibody reactions and all, by we can able to analyze by using this ELISA reader. So you try to get a training for this also. Likewise, gene gun is methods of gene transfer mechanism. It helps to gene transfer mechanism. Then the microarray. So these are the technology we must know. I already told you what all the technologies, hands-on training, the skill that is essential in this field of biotechnology. So without the technical skill, we cannot survive in this field of biotechnology. So that's what I try to say one again and again. I force you to learn techniques. At the same time, you must know the basics and principles of those techniques. Because without principle of those techniques, for example, I already told you the DNA isolation, how to dissolve the chemical substance. We have to find out the solution for dissolving either protein or nucleotides, or sorry, protein or fat or carbohydrates, because cell wall is made up of this three molecules. Okay, we have to use any alkaline solution or enzymatic lysis of the break for enzymes for breaking the bond between the molecules or atoms, or else we can go for homogenization, physical denaturation of the cells. Physically, we can able to lyse the cell by using homogenization. So likewise, we have to think our own innovative ideas. Even if it is not available in this linear procedure, linear manual or protocol, we can go for your own protocol. That's not an issue. But if you want to go for your own protocol or procedures, mean you must know the basics. What's happening? What's there inside the cell, or whatever it is. Okay. If you understand the basics of the practical or particular things, we can design you can design your own protocols. Okay, in case of sodium hydroxide is not there, you can try sodium chloride. But you are not supposed to do all these things without your guide. I already told you, once again I told you, without guide, without proper guidance, without experience, you should not supposed to do these things. At the same time, the innovations are comes from this kind of random checking. That mean random try. You have to try this kind of uh, other you have to find out the new way of approach. You have to find out new idea or innovation of the methods for your practicals. Okay, so usually the students, what they used to do in our college life or school life or whatever it is, they used to do mugging. Okay, but some people has inborn talent as memorizing capacity. Even they read it once, but for others it very hard. To be frank, I'm very bad memory power, but some people have very good memory power. But don't be bookworm. But you should not be bookworm. Think innovative and learn all these things practically. Without practical knowledge, the theoretical knowledge is not enough to survive here, because industrial aspects nowadays, what are the practicals we are doing in our laboratory, in our institutions, that's totally differ from the industry. They never do all these things. So simply they send the chemical substance. In case of the Thyrocare, one of the best example, Thyrocare laboratory in Mumbai, one of the Coimbatore. person is having the Thyrocal. He's the CEO of the Thyrocal. He's the owner of the Thyrocal. But there are thousands, millions of testing samples are tested by each and every day. They simply send the sample inside the machine. They'll give all the results: protein, protein color, protein, vitamins, minerals, blood cell. Every results will come within a fraction of seconds. We need not to, no need to go for this kind of um, traditional older technology and all. Okay, because it was updated so much. Simply, if you are submitting the sending the samples, blood sample or urine sample or motion sample or whatever it is, just to put inside the machine, they will give all the results in internet. So anywhere anyone can take the results. They can operate the instruments also. Even it's maybe a Mumbai or Delhi or US or UK, you can able to operate the instruments from here onwards. We can get the results from here onwards. That kind of instrument technologies are developed right now. But even though the basics are important, already once again I tell you the basics are important for knowing this kind of techniques and all. So practical experience is very mandatory. When you practically learn something, you don't need to cram or stress your brain. Okay, it's easy to understand by doing the practical experience. When you learn practically, then you can save the time instead of memorizing or mugging up. Yeah, of course. Once we saw the movie, say 20 years back, I can able to, if I am asking the questions from that movie, you can able to answer. But if you are reading some stories or if you are mugging up some materials for the 10 years back or 2 months back, you cannot able to answer if you are memorizing the concept. So don't try to memorize yourself. That develop a better understanding. Practical experience gives better understanding. Just by reading concept or statement will not help you to develop your knowledge, even though it may explained in a good manner. You can understand it only by applying the concept practical manner. So what are the things you are reading in a laboratory is not helpful for you. So if without doing a practical manner, and if you cannot forget by yourself, even if you try, so try to understand the concept to get good skill instead of mugging up. Okay, it's all my humble request to students. I don't blame you. So everyone is, even myself as a student, I do the same mistakes. That's what I tell you. So you improve the skills. Training, exercise, innovation are must to improve your skills. It could not come out through theoretical knowledge. Simply, the books will not help to do things practically. Hearing and reading some other education news in India will help you to know about all things which happening all around the world apart from the book. And practically, by using technically, if you're doing, it was very interesting, and also is the very easiest one. In the school days, we have learned something in our chemistry lab. It's some colorful salt solution which makes us more interesting to do, even if they right or wrong. Because it's all happen in our life also. But for this pandemic situation, you might not have the real hands-on training in your laboratory because lack of timing, lack of this lockdown period. So try to get some experience in the internship or training program or some other or in a laboratory. Okay, don't try to get theoretical knowledge and get the certificates from as a biotechnologist. Don't claim as a biotechnologist by using your theoretical knowledge. Try to expose yourself. But if some students like geography means they just studies the weather conditions in book only. They never go for hill station to view the climate changes. Yeah, of course, we never don't try this kind of changes and all. While practically knowing about something, it makes us to understand easily and too interesting. So these are the things I just want to share with you and can motivate the teamwork, because while doing the research, the teamwork is very essential. While doing the practical session, we can do it team and share our knowledge with friends and will enhance our skills high. You can practice it's as a fun to each other company and they're no boring. You can able to know about teamwork and coordinations. So teamwork is essential each and every research and all. So finally, I just want to tell you one thing: the practical versus research. There are slight difference between these two and all. Okay, for an example, I'll tell you. If you want to do if you know the blood grouping is one of the simplest methodology, simplest practical or simple techniques for identify the blood typing, blood group typing, uh, for any kind of people. So, okay, so the thing that I'm going to talk about the research as well as the practical. So practically you know how to do it, but you don't know how to make it as a research. If you are doing blood grouping for 10 people or 20 people or 50 people, take and consider as at 100 people, 100 you are just you are you are finding the blood grouping for 100 people. So we have to get the results. So after the blood grouping, if you are getting the results, we have to tell the result. We have to tell uh, the results in different manner based on the sex. What the for example, I'll tell you, in case of out of 100 people, the 60 people are having B positive. So we have to mention the B positive is very high. We have to make a bar diagram. Okay, B positive, the number of person total sample size is 100. Out of the 100 sample, 100% of the samples, the 60% of the people are having B positive. So B positive is universally is very high. So as well as the very lowest blood grouping, it's maybe an AB negative. AB negative is an you can make a graph, you can able to, or you can go for bar diagram, you can come for the conclusion, and you can able to differentiate and classify the results in sex wise or male or female, or blood group aspects, yeah, or mm, how can I say, blood group or age or sex or so many things we can able to differentiate. We can give the same result. We are having only one results, but we can be able to give the results in different manner. That that kind of experience we must have. So that's the research. Like, research is not so much of difficult. uh, don't think okay, we are UG students, we are not researchers. Don't think like that. You are also a scientist, you are also researchers. The thing is, you have to understand why we have to do what we are doing, to how to interpret the results as a research. Okay, that's the interpretation of the results, that is called as research. Okay, we must know what is meant by interpretation. That is one of the things we must learn from your faculty or your experience, or reading research articles or review articles, etc. Okay, so first you try to read review articles and research articles in your field of choice. உங்களுக்கு பிடிச்ச ஃபீல்டில் you try to read some research articles based on by your guidance of the faculty members or your guide. You first you have to choose your own guide. Okay, based on your choice of interest, or based on choice of research, you select your guide, and then you go for reading the research articles, reading the review articles. Then you start writing your review articles. Review articles is nothing, just if you want to review one movie. For example, you know, all of you know very well about the review of the film. For example, if you want to review about the for Bahubali, if you want to review the Bahubali mean, we have to read at least, we have to see at least thousands of films similar to that Bahubali, from the Ramayana and Mahabharata onwards. Okay, then only we can able to review about this movie, this Bahubali, without seeing all those movies. This is if I'm first time I'm watching this movie in my life, Abhina, I cannot able to go and review the movie. I should have the experience in that particular aspects. I don't need to be a director. I don't need to be a producer. I can able to, I'm an audience, but I can be in my experience I can see lot of movies related to this Bahubali, from the Mahabharata and Ramayana and Indian Korean movies and South Asian movies and American movies, Russian movies, all these movies. I should have this kind of experience. I can, after that I can able to review the frame by frame about the Bahubali. Okay, that's what I'm trying to say in this. If you want to review all the articles, or you can able to start writing your review articles from as a UG students also. But for that, we have to work hard. We have to read at least the thousands of, at least over hundreds of research articles. Okay, you try to read research articles, then you can become a full scientist. Okay, don't underestimate as a student. I'm a BSc student, so how can I write the research articles and proposals, or review articles and all? Okay, it's all nothing. As a student, you can do all these things. Okay, try to do yourself. Try to get the knowledge from your faculty members, your teachers, or your guide or mentor, whatever it may be. Oh, okay, even myself. You can call me at any times for getting this idea, this kind of what kind of ideas I can do, my research or project or internship or paper writing, review writing, anything. Okay, so this is about my talk. So I hope you all understand. I hope this 1 hour is meaningful in your life. And I thank all the management people, as well as the faculty principal, HOD ma'am, as well as Dr. Anjani, and my dear students and my dear participants. Thank you once again all. I come up with the end of the session. So if you have any clarification or doubt, you can ask me anytime. I share my number also. I'll mail ID. You can get my mail ID from by the Google. Just put a Prakash Vels University. You can get my details also. So keep in touch with me. I wish you all the best. Thank you.
Thank you so much for your valuable time
Madam.
for us, sir. Hope everyone learned new things from your session. We are very thankful to you. We have question and answer session now. Those who have questions and doubt, you can put a message in the chat box. We'll convey the questions to the speaker.
Yeah, please.
Yes, sir. We have few questions from a participant, sir. That is Dash. He has been asked that what are the biotechniques used in this pandemic situation to identify coronavirus in laboratory test?
Yeah, of course, RT-PCR. Everyone know this real-time PCR, as well as another term we used to call as reverse transcriptase polymerase chain reactions, because COVID virus is a RNA virus, so we have to convert as a cDNA. Once we've converted the cDNA, then only we can go for amplification. So RT-PCR as a real-time polymerase chain reactions, this is the one of the best method, and 100% we can able to give the results. But even though we have some kind of false positive, false negative results we are getting now due to wrong amplification, because we cannot able to trust always the results. Okay, because you as a technologist, as a technologist, we must know this term: false positive and false negative. These are the two things we use. It's used to happen in clinical industries, because sometimes it may gives the negative results, but fortunately they may get the positive result. Sometimes they get the negative result, but in our results they may get positive results. So this may happen everywhere. So as by the experience you can able to differentiate whether it is a false or right or wrong. The things we have to understand by using the things. um, of course the real time is one of the best method right now. We don't have any other option best.
Okay, sir. Now the question is from Ram Kumar. That is, what are the important biotechniques needed for biotechnology jobs?
Actually, that's why I tell you it's based on your interest, because nowadays the cancer research is very much high. Because wherever you go in India and abroad, if you want to go postdoc in abroad in US, the best place of doing PhD or postdoctoral research in me as the best choice is the United States of America, of course. The most of the research is going in the field of cancer. So flow cytometry is a cell analysis. They differentiate the flow cytometry. It's one of the best technology to differentiate the dead cells, live cells, and damaged cells and all. Because cancer is one of the how can you say, it's, it's a very big problem for the society. So, but we should not think like that. Even the environmental studies, even the for example, if you want to go for food industry they are getting a good job. Good job mean simply we can able to get lakhs and lakhs salary as a quality controller. For the quality controller we just need a isolation and identification of the microorganism from the sample. This we must know the basic microbiological test is enough for that food industry, because they simply identify the micro prevalence of the microorganism, microbial contaminations or microbial load in the food industry. For example, Reliance, Hudson, crew industry, Amul, whatever, wherever it is. They simply take the small piece of small quantity of the fruits or vegetables or ice cream or milk or whatever it is. They analyze the microbial test. That's it. That is the role of a quality analyzer as a microbiologist uh, until their retirement. So, you just know the difference between this flow cytometry, one of the costliest instruments in this world. It may be 4 or 5 crores, I think so, in India itself. In foreign and all, I am not sure. I know about the cost of the instruments and maintenance of the instruments, but it's another side is one of the cheapest method in these techniques, microbiological analysis. It's just petty plates and medium and laminar flow chambers is enough. Just small skills enough for this kind of microbial analysis. But even the both of you getting the equal salary. Even in India, you can get a lot of jobs in both side, as well as in US or UK or German, France, wherever you go. Food industries, a lot of food industries are there. A lot of cancer disease is going on. Compared to cancer research, flow cytometry, the numbers, the ratio is very high in this food industry. As a QC, there are a lot of openings are there, but they require only the basic preliminary skills, basic microbial analysis. If you're strong in the basic microbial analysis, you will get the job, or you can do a lot of research. You can do so. We cannot able to classify which techniques is suitable for me to get a job and all, because as a biochemist, I already I told you that as a biochemist, within they just estimate the protein level or carbohydrate level or protein vitamins level in particular food products. They used to analyze this kind of things from the food industries. So what I mean is, we cannot able to which one is the important or like that. Okay, and because research is very limited, but industry production level is very high, huge industries are there. So that's what. Okay. So try to, you focused on your own interest. Okay, you will get the job surely. I'm sure.
Yes, sir. The next question is from Saranya, that is how we get internship opportunities from manufacturer companies, sir.
You have to approach. You have to get the website. You find first of all, you just get the list of industries in and around your location, and that so many factors are there. As a teacher, I know parents' economic status, as is the location. In case of male or female, they may not allow to go long distance. They may not go abroad. So there are a lot of factors are there. From the beginning, I'll state the starts from our own city, Chennai city. First, you must know what all the industries are there, what all the food industries are there, what all the pharmaceutical industries are there. First, you have to choose your choice of interest, whether you are interested in pharmaceutical line or food industry line or research line or cancer or medical or IVF or so many fields are there in this field of biotechnology. So first you have to choose any one of that, and then you have to find out what all the industries are there in and around the Chennai and in and around your native place or your location. Then you have to get the website address and you have to contact the HR or MD or CEO, whoever it may be. The website itself, you can contact them and you can go for approach through the proper channel. Either you can go through the HOD or principal or proper channel. If you're going, they will offer as an internship. They will give you the internship. So there are 3 4 types of internship we can do. First, the basic trainings modules, because basic fundamentals you can able to go for private laboratory, or real time industries. If you want, you can go for industry. Real time research, if you want, you can go for research institutions. There are different sectors are there, so you have to choose and you have to contact yourself. Don't think they never of course, if you are sending the mail for 10 people or 1,000 people, you may you may receive only few reply from those companies. My side, a lot of food industries, they're offering free internship. Of course, they don't collect any charges. A lot of government sectors also, they are offering free internship. The government, they are Indian Institute of Science and Academy, IASC. They are offering fellowship, where the internship with the fellowship. They are giving the best offer for you. Each and every year they used to call for. You can apply and you can get the internship training program, as well as private companies also giving a lot of. But you have to choose. You have to Google all these things. You have to find the places You have to get the contact details from the Google. You have to approach them through the mail, through the phone. Okay, don't try to go there directly. Okay, if you are going directly, they don't give the response to you mostly because they are busy with some other work. You try to contact through the proper channel with the email through the mail or contact number. This is the right way of approach.
We'll go with the last question, sir. How coronavirus is processed in RT-PCR, sir?
Coronavirus, of course, the real-time PCR, reverse transcripted PCR. First, they used to isolate the RNA from the virus. Then after that, they is convert as a cDNA. Is already told you, complementary double-stranded nucleic acids. Then they will go for denaturation or leveling temperature. The normal PCR procedures. It's a simple process nowadays. Everything is available. um, it's a ready-made mixture is available in laboratory. Like for example, for the PCR analysis, we need small basic chemicals. First is template DNA. Of course, the viral DNA is a template. So they used to collect the blood, then go for um, bloods via separate the RNA isolation, then go for PCR analysis. For the PCR, they require a template DNA, a template that means RNA viral DNA, as well as TNTP mixture. This is a master mixture. We used to call this master mixture. The DNTP is contain deoxynucleotide triphosphate, ATGCP, deoxynucleotide triphosphate, mean DATP, DGCP, DGTP, DTTP, deoxy tyrosine family triphosphate. Okay, all the components are there, as well as the Taq polymerase. Taq polymerase is one of the enzymes which helps to amplify the molecules. The next one is the primer, yeah, specific primer for that particular organisms. They used to synthesize the specific primer. That primers only amplify the particular viral gene, any one of the genes, but specifically located only in the viral COVID virus. Okay, that kind of primer we have to synthesize uh, after the sequencing of the whole complete genome sequencing. First, we have to go for before going for this RT-PCR. First time, why we have delayed for this RT-PCR testing? First, initially we have to go for sequencing, complete genome sequencing. We must know what is meant by genome sequencing. For example, genome mapping, we used to call this genome mapping. You try to read articles, but it'll take long time. So it's first, after the complete genome mapping or sequencing, we have to identify the specific primer for the specific amplification of that particular viral DNA, viral RNA. By using that specific primer, we can go for amplification. So that's after doing the amplification only, I told you already, sometimes we can able to get false positive or false negative results, because sometimes the primers may go and bind somewhere else. That is called a strong amplification. Okay, but the product size will be vary from each other. நமக்கு கிடைக்கக்கூடிய ப்ராடக்ட் சைஸ் வந்து it should be a same size. Okay, in case of changes in the size of the product, that means amplified products, based on that we can able to differentiate whether it's a false positive or false negative and all. So this is the procedure protocols we used in our RT-PCR testing laboratory. By YouTube you can see all these things. Okay, you can get all the idea about the RT-PCR. As a students, you must know, you must be strong in this RT-PCR and all. Okay guys, thank you. Any more questions are there?
All the other questions will be answered. Thank you so much for answering all the questions, sir.
Thank you, ma'am. Thank you so much.
The attendance link will be posted in the chat box. Kindly fill it. If not, barcode has been shared in the screen at the end of the session. You can just scan it. It will be directly go to the attendance sheet. Kindly answer the attendance without fail. We came to the end of our session. Vote of thanks will be given by Saran Kumar, third year Department of Biotechnology.
A very good morning to all. It gives me an immense pleasure to deliver the vote of thanks to this event. I would like to thank our chief guest and eminent speaker, Dr. B. Prakash, sir, who honored this function with his inspirational thoughts. And also, I would like to thank our beloved HOD, Dr. Rajeshwari Hari ma'am, and also Ms. Sandhya ma'am and Shanmugapriya ma'am for their great effort, and also our volunteers. And last but not least, our beloved participants for making this session very interactive and meaningful. Once again, I thank one and all who present here. Thank you.
Thank you, Saran. Thank you all for your interactive participation. We'll get back to you with another event. Stay home, stay safe. Thank you.